Well, thank you very much to the organizers for the invitation to come speak. Thanks for everyone for joining us this afternoon. So I'm going to walk you through some work that's occurred in my lab that is happened a bit on Earth, just a bit above Earth, and also some plans for what's coming soon for some missions to the Moon and to Mars. And I'll start first, of course, with conflicts. I have a number of conflicts with most of the people in the industry booths in that room over there, but a lot of these people are collaborators, colleagues with whom I work to get some of this technology out into space. And so I'll also close a little bit with a discussion of colossal at the end, which will touch a bit on de-extinction. So first, I want to start with just the facts of where we are today, that more than ever, we are a spacefaring species. If you look at since 1957, when the Sputnik first launched into space, most of the objects were either Russia or United States derived that were going up into space, and you can see there was competition for many years. The Cold War was the first space race, really. But something happened in the past few years. Actually, this one became from an increasing chart to a super exponential increase in the number of objects and crews launched into space based on the United Nations data. And so actually last year set the record for the most number of crews and objects launched into space, including some samples from the UK Space Agency, so really driving things forward. And there's even now architectural firms designing moon habitats with ESA. So if you want to be if you have any children and you think, what can you be when you grow up? You can legitimately say, I want to be a lunar architect. That is an actual job that exists today. Here's one of the firms that is doing that today. So it's very exciting. And even plants have been grown on the moon. This is from the Chang 4. We're taking Earth-based soil and sending cotton plants on the moon. You can see these are plants that are growing on the moon. You cannot yet buy cotton pants from the moon, but that may come sometime soon. We also have seen that actually even with lunar soil, you can take modified Arabidopsis from Rob Furrell's work and actually get them to grow as well. So we can even tweak the microbiome as well as the plant genome itself to get them to grow in lunar soil. And so it's really an exciting time. It's kind of the new space race. Even China is planning to get there by the year 2033. And NASA and other space agencies are also thinking about getting to the red planet. So this raises this question of everyone wants to go to the moon and then to Mars, but what will it take for us to inhabit Mars? How can we safely arrive there? How can we learn about the body and genetics and sort of the precision medicine of spaceflight, essentially? So I want to review some of the work that's occurred in my lab in the past few years, and then what's come of some unpublished data in more recent missions. And I'm going to close today with a bit of a philosophical note about why we do this work and why I think it's important. So I'll first start with one of the first studies that we got to work on with NASA, which was the NASA twin study. And some of this is published, so I'll go through it quickly. But basically, Mark and Scott Kelly, identical twins. Scott went into space for 1 year, and we had the chance to get blood and stool and urine, saliva from him during the mission. And actually, later on, his brother, Mark Kelly, he became Senator Kelly. So we have the beginning of the senatorial genome project in my freezers in my laboratory. So we could maybe start to do a GWAS of Republicans versus Democrats. And so, for example, could you CRISPR a Democrat into a Republican or CRISPR a Republican into a Democrat? I put this into DALL-E for the AI just to see what it would come up with. This is the image it came up with in DALL-E, or Stable Diffusion, just to see. And you might think it's a little bit of a political sidebar, but I'm not the first person to think of this. You may remember this paper where it looked at, well, who voted for Brexit? And was there a reason on chromosome 3, these alleles here, in theory, are associated with people who voted for Brexit? So could you CRISPR those loci and change their vote? Of course not. Voting is more complicated than that. And of course, this represents more subtle population stratification of urban and rural populations. But it is interesting to see there are some differences already published. Anyway, that's the end of my sidebar. Anyway, back to the mission. He went up into space, went up there for a year, and took these beautiful photos as he was up in space, charting. I wish all of my flight subjects and test subjects would tweet all the time and put on Instagram what they're doing. He's doing some juggling in space, you can see here. It's very easy because it stays there, so it looks complicated. He also grew a zinnia flower in space and had all this great work in, again, blood, stool, saliva, many collections that we got back from the space station. But after 1 year in space, he made his way back down to Earth, and this is actually the video just released of the first time he tried to walk, again, after not using his body, really his muscles that much with any strain over the course of a year. He came back. This is his first time trying to walk very shortly after landing. And you can see he can move, but he's stumbling a bit and started to also have a bit of a reaction. He actually had some stress, some strain, and when he got back to Houston, his skin started to break out in this rash and discoloration anywhere that he had any contact. Even the weight of clothing itself would make his arms break out into a rash. And he had flu-like symptoms. He wanted to go to the emergency room. He had to mostly spend some time naked just to avoid getting any rash. Some people do that for recreational purposes. he was doing it for medical purposes, right? So he had to, just everything hurt basically in his body. And the real question we had is just why? What happened inside his body after a year in space with the radiation, the change in fluids? What occurred in his body, and could we learn from this mission and apply it to other missions? So we deployed a multiomics analysis of human spaceflight to better understand this. And again, I'll summarize quickly some of the highlights, but we looked across many modalities of biology, so microbiome, gene expression, metabolites, cognitive traits, protein changes. And one thing that was really surprising is when clusters of biology would move in concert. So we could see some of the modalities of biology indicating co-regulation. We could see, for example, many things increased in flight, but then would come back down. Telomeres, for example, got higher in flight, but did come back down. And so this led to some headlines that indicated that Scott Kelly was getting closer to the fountain of youth in space, but it did come back down to Earth. But that was just with qPCR. So since it is a genomics conference, we of course wanted to use some of the more longer reads like Nanopore and PacBio. So we also developed an algorithm called EdgeCase that could take long reads and map to the extended reference genome and basically count on a base level resolution the telomere length. And we could actually see here not only the length, but also some of the motifs, the TTA, GGG motifs. You can see here these spikes of color where you have the mapping of the reads, and then the spikes of where you see the density of the motifs. And you can see the canonical motifs in green, but we did pick up other motifs. And you can see here with the Oxford Nanopore, we got very, very good data with PacBio as well. You can see we can get this burst of the density of when you hit the telomere, it's very clear. And so we confirmed with the long reads as well and showed that on average, he got longer telomeres in space. You can see on the left is Scott, but he did also get some shorter telomeres on average. He did get irradiated, and I'll come back to that later, which we could see. And we published this also as the algorithm as well as the results in Genome Research just about a year and a half ago. So the other thing I really like about this paper, though, is we're looking at Scott Kelly, but first, of course, we looked at controls just to get a sense of the method and how we're doing, and looking at genome metabolic controls just to see that we could map the reads correctly. In an Australian Jewish trio and a Chinese trio, you can see these bursts of color, which are the non-canonical telomeric motifs showing up at the end of the telomere. So telomeres are between 5 and 8 kb. You can see a lot of them on average, but you can see there are population-specific non-canonical variants showing up, which is like most genetic variation. It's usually ancestry-specific and heritable, and it's the same thing in telomeres. There's these hidden little variants hiding on the subtelomeric and telomeric regions, a lot more than we can now see because of work like the Telomere Consortium, where we can map these reads, and we can see it across all chromosomes, these bursts of colors and these hiding variants floating in basically the subtelomeric and telomeric regions, even some CPG islands as well. So that's the telomeres. The other thing we looked at in this mission was what happens for gene expression. Of course, many, many genes change expression. You can see bursts of orange and purple, or genes going up and down, we can see. And we could see enrichment for the immune system, DNA repair, and hypercapnia genes. And what's interesting about this is that he wrote a book about his year in space, which I wish every subject from every clinical trial wrote a book about it, because we could look for things like, did he have hypercapnia genes? And he did. And then we asked him, well, what was the carbon dioxide level? Because it's too much carbon dioxide or oxygen levels, they were actually pretty stable. And talking to other crews, it turns out the very likely reason why we think people are seeing hypercapnia is more because of just how things move in space. You can see that air does not move the same way it does on Earth. You can see if you blow bubbles in space, that can sometimes stay right in front of your space. So you get these small clouds of carbon dioxide that can essentially give you a little bit of hypercapnia, and we can see some of that reflected actually in the blood work. So the other thing we looked at in the blood work was cell-free DNA sequencing. We wanted to see basically, what do we see for the expected tissues of origin? When you sequence all the cell-free DNA or RNA in your blood, you get a real whole body molecular scan of which cells are dying or lysing or under stress. And we could see a lot of the hematopoietic system showing up on top as expected, but we could see bursts of detection of other tissues sometimes showing up, like retinal tissue, which is also related to some of the eye damage that astronauts get. So this is something we're doing now for future missions, is to sequence all the cell-free DNA as well. But the one big surprise we got with cell-free DNA was if you look across all chromosomes, it was mostly flat before, during, and after the mission, except for one chromosome, which is the mitochondrial alignment here, which burst up in flight. And what's interesting is there's like basically this burst of mitochondrial DNA we confirm with qPCR, and we've always been wondering about this, but data that's under review right now, we can see from 6 other Japanese astronauts, they see the same thing. So there's this burst of mitochondrial DNA when you get up into space that seems to eventually taper down, but is this a reflection of the stress of spaceflight. And so it's interesting. We think what's happening is this is basically an interferogenic response. White blood cells, when they're stressed, can eject their mitochondria as a way to kind of prime the immune system, is what we think is happening here, is some of the confusion of the body. So to go back to the rash, though, we got this rash. The other thing we looked at was cytokines, what spiked up when he landed on Earth. And this was IL-10, IL-6, IL-1 receptor antagonists. These are the cytokines that spike the most, these pro and anti-inflammatory cytokines. Basically, his body's struggling to get back to Earth, back to sort of a regular existence. And you can see these spikes is probably what led to a lot of the rash. But spikes of IL-6 can be problematic for other purposes. So if some of you in the audience are probably studying clonal hematopoiesis, which is when you start to have an enrichment of mutated clones in your bone marrow that propagate at a faster rate and actually can become abundant. You can see them in the blood. And this can lead to a higher risk of atherosclerosis, cardiovascular disease, or leukemia. And so we've been looking at clonal hematopoiesis in the lab for leukemia patients, but we also took the same panel and wanted to look at what happened to Scott, for example, in space. And here we could see his actual clonal hematopoiesis decreased in space. You can see the allele frequency of the mutated TET2 mutations went down in space, kind of the opposite of what we expected. So going to space is so far good for clonal hematopoiesis, which is unexpected, but it did come back when he got back to Earth. So you can see on the right, the allele frequency came back in the months and years following, and we're doing annual blood draws now to keep an eye on this clonal hematopoiesis for this crew as well as other crews with NASA and SpaceX, which I'll come back to. So one other fun fact about this paper, we published this again about a year and a half ago, and you can see here we had to make a summary figure. Like most of you have to make summary figures for your papers. One fun thing though is we made, here's before and after flight and space flight, and they've got lightning for radiation. But if you zoom into these cells, you can see there's a baby Yoda that we hid inside the PDF. So high-resolution PDFs are a great place to hide little Easter eggs. I highly recommend it. But this wasn't totally our idea. We were inspired by someone who did this in 2019, where someone hid a piece of Donald Trump's face in the stool in the unusual aspects to the fecal illustration. If you didn't see this, the journal had to take it down, and they realized that the extract fecal DNA had unusual aspects, and you can see there. So now you can't find it, but it is on Twitter. If you look around, it did happen. It was there. In any case, overall, good news from that mission. We learned a lot from these multiomics analysis, but clearly this was great, but also one of the least powered studies in human history, right? So we clearly need more, and it's more data, and so we launched an IRB and a protocol and a consent form at Cornell. So if any of you are going to space anytime soon, please let me know. We're recruiting people. The first, and what we want to do with this mission is we're calling it the Spaceomics and Medical Atlas Protocol, which layers in a lot of the same molecular tests that I described before for the twin study, but now it's open to any commercial entity in any mission. So this is a lot of cell-free nucleic acid testing, multiomics, epitope profiling, microbiome, and multiomics analysis before, during, and after flight. And I'll present to you what we got for the first chance to use this. Actually, was in 2021, the Inspiration 4 all-civilian crew that SpaceX launched, and so they actually went up higher than the ISS or Hubble, up to about 590 kilometers at apogee, at the highest peak. And of course, when they get up into space, they're excited because they're a crew. They take some selfies because they're excited they're in space. But they did eventually get to work and take actually over 700 swabs. They did blood draws, ultrasounds, a lot of work during the mission. They safely were coming back to Earth. We were very excited. They landed. They splashed down into the ocean, made their way back, and they were, anxious to come back, but they were very excited because they landed and they didn't die. When they got, you can see the very excitement. They made it. They're alive. We were very excited because they were back and they didn't die. They brought other samples. Very excited. We did drink a fireball shot there, which I do not recommend. In any case, we got them back up onto the ship and got to work there. Took the HEPA filters, the swabs, and we started to ask some of our questions for this crew, which we now are doing for future crews. And a lot of these data I'll show you now in review. But one of the first questions we just had is a simple one. Do skin microbiomes get similar in space? They're in a tight Dragon capsule. It's very small. And so do they get more similar? Kind of like what happens during sports. Before and after you play a sport, you'll rub into people, and your microbiome of your skin will become more similar to your opponent after the game. I mean, even if you're in a crowded elevator, your skin microbiome might get a little similar to someone. Depends on who you're riding with. But in any case, we wanted to see if this happened in the space capsule. And so here we sampled 10 areas around the body to see what happened before, during, and after the mission. And then what we could see is, quickly on the right, the diversity decreased of the crew during the mission and after. It was very noisy, but the trend was there. They got, on average, more similar. And we also wanted to see, where do we start to see the most species per body site? It turns out, actually, the armpit is the one that's got the most diversity of species for this crew. And so you might look at your armpit in the shower and think it's a dark cavern of despair and disgust and detriment. But you should not do that. You should look at your armpit as a wonderful rainforest of species and diversity as an opportunity. That's at least what I do now when I'm in the shower, so I'd say. In any case, so that was what happened to the microbiome. We then also wanted to use some of the latest 10x genomics kits for multiome profiling, which give you an open and closed chromatin profile as well as gene expression that's changed on a cell-by-cell basis. So this helps us address which cell populations are the most impacted by spaceflight, and also gives us a really rich immune profile of the crews. So we did, this is a fairly standard protocol now, where you actually can basically isolate single nuclei, pull out the attack and gene expression library, and then sequence them aligned to the human genome, and you can do joint cell calling to get cell type specificity. And we got over 150,000 cells examined, and of course is the obligatory UMAP plot, which I like to call the Rorschach plots for genomics. Like, you can see your future in here, you can see what you want to be, but it's mostly blood cells, T cells, B cells, dendritic cells, and natural killer cells, as expected. But then we wanted to see the difference. So where do we see differentially expressed genes, and where do we see chromatin change by these different cell types? And so you see each row is a different type of cell, and you see genes going up in orange and down in purple. So many more genes were increasing their expression on average. But you can see at the bottom, the number of cell types that had a lot of changes, chromatin, was not all of them. In particular, the monocytes had a much more dramatic change of opening of the chromatin, whereas other cells had much less so of these kinds of changes. So we can see these cells, in particular, CD14 and CD16 monocyte cells, are the ones that are the most stressed and actually generally opening consistently across all the crew members in this data set. It's really interesting to see that there's a very cell type specific response to spaceflight. And then we also want to see, compare the day that they landed versus 6 months later. Do they persist? Do they change? And so we can see a lot of the changes in differentially expressed genes versus differentially accessible regions. Of course, most of them returned back to baseline, but not completely. So even 6 months later, there were still several hundred genes, several hundred or even thousand loci, depending on cell type, where we could see some of the chromatin had still been adjusted and not fully returned back to normal. And so then we want to think, well, we have 2 male and 2 female crew members. Was there any difference based on sex? You know, it's a bit underpowered, but we wanted to at least begin to ask this question. And so far, we could actually see that females have less overall DEGs, and they return to baseline faster. If you look by cell type, the number of up and down regulated genes that take longer to recover, it's mostly the males. So if we're sending crews to the moon and Mars, we should do at least half females, to get back to the opening comments. Probably 60 or 70% females is what I'd recommend based on the differential expression data. And this is actually good because the next release of astronauts going to the moon with the Artemis 2 rocket is going to be at least have 1 female, but I think we should have more based on the gene expression data. So the other thing we looked at based on these genes is what pathway changed. So we look, of course, did an ontology assessment to see what overall groups of genes were changing, and chromatin assembly, epigenetic changes, and telomere stress also increased. And so then we did a quick qPCR to see did they get longer telomeres. Indeed they do. This is work with Susan Bailey, and this is just by qPCR. It's only statistically significant for one of the crew members, but even with just 3 days in flight, we do start to see this burst of a lengthening telomere on average. But then what we're in the middle of doing now is finishing a paper on taking some direct RNA sequencing. So looking at the RNA with the single molecule method of Oxford Nanopore, because here you can actually get full-length transcripts. You can also do this with PacBio, but here we can get not just the long transcripts, and you can see getting 4 or 6 kb transcripts, but also base modifications. We can see methyl-6-adenine in the RNA, and so far we're picking up the DRAC motif as expected across these data, and we're in the middle of finishing this paper now, but we can actually see this pretty well with the direct RNA sequence data as well. So with that what the Inspiration Forward mission completed, what's next is we are putting all these data into NASA's Gene Lab, which is the repository for accessing all the science of anything that's flown up in space, all the omics data, the molecular data, the telemetry data. And we're also launching later in a few months, basically the Cornell Aerospace Medicine Biobank, inspired by the UK Biobank and some of the work from other groups like All of Us, is to make it so you can access the data as well as the samples for any of these missions and building these out. And so this is actually kind of the new biobank. And we just got samples in yesterday from the Axiom 2 mission, which included John Schaffner and Peggy Whitson, and also 2 Saudi astronauts. And what's also coming up in the next 1 to 2 years is Polaris Dawn. Is the first mission will be likely later this year, and there'll be 2 more Polaris missions flying up, and the last one will be the Starship rocket, which can carry as few as 12, as many as 100 people into space at one time in the next basically 2 to 3 years. And so the very first mission we'll be working with the Polaris Dawn crew to actually prepare for this and run these same protocols. Now, some of you probably saw the Starship did first launch a little bit ago. It did explode the first time it went up into space. It had a rapid, unscheduled disassembly, which is what happens to me sometimes on Saturday nights. I'm at the bar, I'm just an unscheduled disassembly. But the fact that it flew, that it got up there, and has that much capacity is extraordinary. So we still think we're on track for the next 2 to 3 years to have Starship going up very soon. And the other thing we just finalized with SpaceX, actually just this past week, was a testing of the Mark 1C that we can fly actually up and has been flight approved. So we're hoping to actually do a blood draw, rapid library prep, sequencing, and get genetics, epigenetics, and telomeres all within about 2 hours in flight, if all goes well. So the protocol is now finalized. So I want to close in the last session of the talk a little bit about, on a bit of a philosophical note, and also ways we can look ahead. And think about not just measuring biology and thinking about associations and differences of space flight or people on Earth or microbiomes in the subways, wherever you might find them, but think about becoming more active and think, can we defend biology? And the reason I think about this is because of the quantified risk we know for astronauts. So, for example, if you look at 8-oxoguanosine, which is a damaged nucleotide you can see in urine, in almost every mission we've looked at over almost 60 astronauts, we consistently see the amount of damaged DNA coming out, being or eluded out of the body, and we can see this spike here for almost every mission we've ever looked at, actually. And this is in low Earth orbit. This is relatively safe as far as radiation. If you compare low Earth orbit, you can see Scott Kelly's there in the sort of light blue is 146 millisieverts of an effective dose. The Inspiration 4 mission was 4.72 millisieverts. You can see, so only 3 days in space, and that's more than getting several dozens and dozens of X-rays. But if you look at going, say, to Mars, going to Mars and back, it would be about 1,200 millisieverts. It would exceed the lifetime recommended maximum dose of radiation for an astronaut because a lot of it's between planets, and there's, of course, there's no molten core for Mars. It stopped spinning a long time ago. There's no Van Allen belts. There's no protective magnetosphere. So you're really, with the farther we travel away from Earth, the more the danger we have. And so this raises questions of what could we pharmacologically protect astronauts, physically protect them, of course, but could we genetically or even internally protect astronauts? Could we, like in what we've described before, is could you engineer a perfect astronaut, or could you, and then even should you do such a thing? What are the ethics of making humans fit for Mars, and what are our options? And this seems a bit controversial and unusual until you think about what's happening clinically. There are, actually at this point, over 1,000 different CAR T trials where we're modifying T cells and fusing them back into patients. This is one of our clinical sites in Shenzhen. And also, we've published a review. There's now actually, again, thousands of different modified cells being infused into patients as we speak. So it's not that uncommon. And actually, some of my favorite, most recent work, there was a session earlier today about this, is some of the work for actually doing somatic CRISPR to treat sickle cell disease or beta thalassemia by reactivating fetal hemoglobin. So actually doing CRISPR actually for a treatment of disease. And you can see here the blue is the spike of fetal hemoglobin, one of my favorite papers of the past couple of years. And that's just the thing about CRISPRing of the genome, which has its problems. But on the fabric of life, we don't have to just modify threads. You could change how the threads are utilized. So you could do epigenetic methods for modifying fabrics of gene regulation. And actually, there's a great review paper by Charlie Gersbach that shows there's now many ways you can actually have a deactivated Cas9 system that still has the homing beacon properties, but bring in with it a TET or a DNA methyltransferase or a histone acetyltransferase. So zoom in and then lock into different loci and epigenetically reprogram what's present. So I have a great postdoc in lab, Jen-Kun Kim, who's been doing just this, actually, taking Cas9 systems to take specific genes that we know we wish were turned back on in flight or turned down. And you can see here this ability we can actually do with some of the SunTag systems, 50 or 25 or 8-fold different increase in levels of expression by targeting specific loci. It's kind of like going into a cell and turning on and off a light switch, depending on which gene you want activated. So this means that we are not only subject to what the genes might be doing natively, you can actually target them and turn them up or turn them down, much like playing a piano. And so that's if you target known genes. To get a little bit, again, out there, because I'm ending on a little bit of a more exploratory note, we have also taken genes from the tardigrade, this cute little water bear. It can actually survive in the vacuum of space. And its inspiration from the Hashimoto paper, when they sequenced the genome in 2016, they found this interesting protein called DSUP, DNA damage suppressor protein, that protects the cells against damage, about 40% reduction. And we've taken some of those proteins, actually made a stable transduced cell. You can see here the DSUP marking in green, DAPI, it's for DNA in blue, and then you see the sites of DNA damage on the right where there's an overlap. And this basically alien protein put into a human cell works well to actually help detect and actually prevent some of the breaks and repair them faster. So we actually get about an 80% reduction in 8-oxoguanosine by having this hybrid chimeric system. And you can see it also essentially is associated with H3K4 trimethylation marks as well. So we think this is a way you can use basically a protein from a different species to help inform how you could treat or even modify human cells to get them more radiation resistant. And I think it really means that all species epigenomes can teach us lessons and give us phenotypes. All genomes, all epigenomes. We can actually take any creature anywhere, and once you have the sequenced genome, it gives you an advantage to more parts of the toolkit of potential genetic treatments, therapies, basically, bits of things you can have in your workshop of a genetics workshop. And so we don't just use tardigrades, we're also looking for other interesting bugs. We have a project in lab looking at extreme microbiome sites or even places where there's large amounts of carbon dioxide, carbon sinks. You can see here, this is Braden Tierney in our lab collecting samples that we found actually one of the best cyanobacteria. So far, it has the record for the fastest absorbing carbon capturing cyanobacteria. And so we think going into strange places in sequencing, not only is it fun, but you also find interesting creatures that can actually probably help mitigate climate change or give us new tools in the genetic tool belt. And so I want to close in the last few minutes on the biggest question of why do all this? And so a lot of times I'll have someone say, well, okay, Chris, the first question people often ask me is, Chris, what's wrong with you? And that's a longer answer. I don't have time for now. But the next question might be, why should I care about spending all this time and money and energy going into space? And it seems like it's a distraction. And I have three answers to that. One is that we can both explore space and solve problems on Earth, like poverty and climate change and systemic issues. We can do both these things at the same time. It is a false dichotomy to say you can only do one or the other. We can definitely do both. In the United States, we passed civil rights legislation as well as got people to the moon in the 1960s, and we can certainly walk and chew gum at the same time as a species today. The second reason I give you is that it is a measure of hope. It is an intangible but essential asset to be able to look into the sky and feel like we are reaching farther than we ever have before, that we have a future ahead of us, that children can dream of a future that is bright. And the third reason is actually maybe the most important reason, is that it is essential to use these tools of technology to discover species, to think about going to other planets, because we know that life is precious. In the past 50 years, about a quarter of all birds in North America have disappeared from climate change as well as habitat destruction. Our modern lifestyle is probably destroying microbes before we can even find them. We're losing species before we've even sequenced them or can characterize them. And Marty Blaser and others have written a lot about this. And of course, this is not the first time. If you look at the history of life on this planet in the last 500 million years, the temperature of the Earth has gone up and down many, many times, right? You can see we've had a world without polar ice caps several times. We've had a world with polar ice caps several times, gone up and down, you can see in blue and orange. But those five markers are that anytime we have a massive and rapid change in the temperature, that's when we see large-scale mass extinction, at least 5 times in the past 500 million years. And this is all, what we're trying to avoid now is increasing the temperature too fast in this era to suddenly create a new mass extinction. And if you take this context, though, it's the last 500 million years, but zoom out a little bit more. 700 million years ago, we had a snowball Earth. And if you take that and put that in the context, basically this last 5, 7, 800 million years of Earth and put in the context of the Earth's history and likely future, we're kind of right about here where we have liquid water on our planet, and it's this very nice but it's very temporary temperature canyon where Earth is. And you can see when I was actually finishing a book about this, and I was thinking in my head, we have 4.5 billion years until the Earth gets engulfed by the sun and is no longer here. I was like, that's a long time. But when I was finishing a book on this subject, it's only in about 1 billion years where the sun will start to get hot enough that it'll boil the oceans and the Earth will probably become uninhabitable. So I was going down in our kitchen, I was kind of moping around my house, and my wife was saying, what's wrong with you? And I said, well, I just thought we had more time. And she laughed because, well, we've got still 4.5 billion years, but the planet. I said, no, but 1 billion years is still a long time, even if it's only 1 billion. I said, yeah, but imagine if you were a patient and you thought you lived to be 100 years old and someone said, oh, you're going to be dead at 20, you'd be devastated, right? So I feel like if you have a time-independent view of humanity, it is devastating to think about that loss of time, or at least that's what I think. So, and I think it's not just humans, though. If you think about everything that's at risk, we always think about the food chain as these consumers, producers, and decomposers, right? These three kinds of life. But I think this misses something. There's actually a fourth kind of life, the only kind of life that's aware of the ecosystem, aware of the frailty of life. Quite literally, something only one kind of species can guard against the loss of habitats. Quite literally, the guardians of the galaxy, I think is what I'd say. But really, only humans are aware of extinction. Only we have the capacity to understand it, even do something about it. And I think that gives us a duty towards all life, present and future, and potentially in the past. And this is some work we've done with George Church and Colossal and others and Ben Lamb, is could you bring back the woolly mammoth? Should you bring back the woolly mammoth or the dodo or other species that are under works? And it is an ongoing project because anytime you take away a species from an ecosystem, it could be just as devastating to put one back in. So it will be done very slowly and very carefully, but it is a very active project as we speak. And I think it's not just for whim or for fancy. It actually, the technology being built will help us a lot with fertilization, with better understanding of embryos, like we just heard in the wonderful previous talk. talk, and also to preserve species. So using technology as basically a manifestation of our duty to all life, maybe even AI as a life form for the next 500 years. This is the shameless self-plug of my book at the end where I talk about this in detail. So with that, I want to thank everyone in my lab who makes this work possible. I really could not stand up here without having fabulous people in the lab. This is an older photo. This is a more recent photo where everyone was celebrating at a Christmas party, and I'm very honored and grateful to get to work with such amazing scientists. Of course, I want to thank funding from NASA, SpaceX, World Quant, and the Gates Foundation, and many other collaborators, colleagues, friends who've made this work possible, especially industry collaborators who made for some of the sequencing. And finally, thanks again to the conference, to the patients, their families, and to everything they leave behind for the next generation. Thank you very much. And I think I'm out of time. There's 1 minute left of time.
Yeah.
But we could also catch up on it.
Yeah. Thank you, Chris.
Thank you. Thank you.
The second plenary speaker is Peter Wisher. He's professor of quantitative genetics at the University of Queensland, and his research focuses on the causes and consequences of human trait variation, and he is a man behind the concept of polygenic risk scores. So please join me in welcoming Peter on the podium.